1 billion adults are obese, and estimates suggest 50% of the global adult population will be overweight by 2030, only 5 years away. Obesity is a life-limiting condition, dramatically increasing a person's risk of developing serious health conditions, including type 2 diabetes, heart disease, and some cancers, and is estimated to cost the UK around £75 billion each year in direct healthcare costs, as well as broader costs to the individual and wider society. These numbers are astronomical, so there's an urgent need to develop new strategies to address this problem. Only 2% of people with obesity are medically treated, and the lack of available treatments may be partially because we don't fully understand the mechanistic details of obesity and its related conditions. It's only relatively recently that we've begun to recognise adipose or fat tissue as a dynamic organ comprising several cell types and not just as an energy store. We need new tools to understand and pick apart these complex signalling networks. However, in the lab, we typically work with overly simplistic and artificial 2D cell models, or incredibly complicated animal models which lack relevance to human disease. To help bridge this gap, in my PhD, I have developed and extensively characterised a new 3D adipose spheroid model, essentially a ball of fat, which allows us to study complex processes in a more physiologically relevant system. To do this, I grow human adipocytes, or fat cells, in dishes which have been treated with a coating, allowing the cells to stick to each other rather than to the dish. This means that the cells will behave more like the cells in your body than cells stuck to plastic in the lab. I have shown that these spheroids accumulate lipid or fat droplets, as shown in these images. They express adipogenic genes and proteins and crucially function like adipocytes. I've optimised assays to measure glucose uptake, where cells take up excess energy from the environment to store, and lipolysis, when cells release this stored energy for use. I've recently been able to use this model to begin to pick apart these complex biology and metabolic disease, and for example, by exploring the effects of inflammation on fat gene expression and function, and looking at specific proteins which may become new targets to treat these diseases. This system therefore provides a new tool which allows researchers to better understand complex and understudied processes in obesity and metabolic disease, and could pave the way for discovery of new therapeutic targets and more effective treatments. As the obesity crisis continues to grow, innovative approaches like this will be essential in improving our understanding and ultimately improving lives. Thank you.